Good morning, everyone, and welcome to this new TMS seminar. Today, the Journal Club Biotechnology team, composed by Michela Ciocca, Loredana Parisi, Ilaria Di Carlo, Giorgia Nicoli, and Camilla Pizzuti, will present this paper to you. The idea comes from the Nobel Prize for Chemistry in 2020, which saw as a protagonist two researchers and their discovery of the CRISPR-Cas9 system. However, this method is not always controllable and could create some problems in DNA modification. And in this short presentation today, we will show you one of the methods that could make this technology more controllable. Enjoy this work and good vision.
Okay, good morning everybody, and welcome to this new TMS seminar. Today I will talk to you about the technology of gene editing that many of you have already heard, called CRISPR-Cas. Nowadays we hear a lot about genetic editings, especially in the agri-food sector, in animal husbandry, in the development of new medical therapies, and the creation of new drugs, or even in the creation of new fuels. But how is it possible to introduce a genetic modification in DNA? Probably some of you have already heard of CRISPR-Cas, especially in this year in which these two women, Dr. Emmanuel Charpentier and Dr. Geneviève Donda, have won the Nobel Prize for Chemistry for the discovery of this system and for its application in gene editing. CRISPR system already exists in nature. In fact, many bacteria use this mechanism as their immune system, which allows them to trace viral DNA and destroy it. To understand what it is, let's think about the name. CRISP is the acronym of Cluster Regular Interspaced Short Palindromic Repeats. It is a non-coding DNA sequence containing short fragments repeated several times, useful for storing viral DNA as a memory once the bacterium has already met it. The second actor is a protein called CRISPR-associated protein or Cas, which thanks to this memory can recognize viral DNA, cut and inactivate it like a scissor. More in detail, when a virus invades a bacterium, Cas protein cut out a segment of viral DNA to stitch into bacterium CRISPR region, collecting a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA that bind a protein called Cas9. The resulting complexes act like scouts that latching on the free-floating genetic material and searching for a match to the virus. So, if the virus infects again, this complex can recognize it immediately and Cas9 can destroy the viral DNA. When researchers edit a genome using the CRISPR-Cas genetic scissor, they artificially construct a single guide RNA called sgRNA, which matches the DNA code where the cut is to be made. So this single guide RNA acts like the complementary RNA to the viral DNA mentioned before, and it forms a complex with the Cas9 and detects the scissor to the precise location in the genome where the cut will be made. Once the cut was made, the repair can take place in two different ways. By the non-homologous end join, which is another repair mechanism that allows the gene reparation by small insertion or deletion, or by homology direct repair that, using a donor as a template, allows to insert a piece of DNA useful for the gene correction or for insertion by a sister recombination. But there is a problem. CRISPR-Cas are uncontrollable, capable of forming double-strand breaks at a specific location where the single guide RNA binds within the genome. So, how we handle this technique? In this paper, they try to solve this problem by CRISPR method that allows the space-time control of DNA ruptures caused by CRISPR. The mechanism behind this method is through the creation of single guide RNAs that are chemically synthesized by incorporate photocleavable residue in the structure. so that these single-guide RNAs can be fragmented in response to light. In contrast, as you can see in the image, normal guide RNA do not fragment in response to light. In this way, therefore, under light stimulus, it is possible to turn off the crisp cutting activity by cleaving the single guide RNAs. More in detail, this cleavage is possible by replacing normal nucleotides with the molecule containing O-nitrobenzyl groups at definite position. This molecule is photocleavable when subject to wavelengths between 345 and 385 nanometers, and thus allowing the fragmentation of a single guide RNA and its consequent inactivation. They use a different single guide RNA by inserting the photocleavable construct in different positions. So then they expose them to the light source and analyze them on a fragment analyzer. What has been possible to observe is that all these single guide RNA have been fragmented. And for the same cutting position was also evaluated the presence of editing of 3 genes that are DNMT1, FANCF, and VEGFA. So, as can be observed, the substitution of the nucleotides with the photocleavable molecule abolished the editing efficiency for some loci, like for example the 22 locus, while for others there is a still high percentage of editing, like for example 57 locus and 74. So, as you can see in this image, based on the result obtained, they decide to create a single guide RNA for the CRISPR-off, inserting the photocleavable construct in 57 and 74 positions. And they call it dual Brex single guide RNA, or DB-sgRNA. To test if it really works, they use the mass spectrometry analysis of the full length of double-strand single guide RNA and the fragmented one. So, as you can see, the peaks reflect the different type of fragmentation in one of the 2 positions or in both. In this experiment, they compared the ability of a single guide RNA to the ones of DB single guide RNA in cutting a product under different conditions. In the first line, you can see a band that represents the full-length product without cutting. When they put Cas9 and the single guide RNA or DB single guide RNA only, you can see here, as you can see here, by the 3 bands, the cutting of the product take place. But something different happens when they add the light source to the reaction. Indeed, the cutting takes place only with the single guide RNA, but not with the DB single guide RNA. So, the analysis of the DNA fragments shows that DB single guide RNA maintain an in vitro activity comparable to the standard single guide RNA only when they are not treated with light. At this point, they decide to start with the in vitro experiments. The first step was to test if the exposition to the light using 345 nanometer long passive filter could influence the viability of the cells. And as you can see in the left graph, the light didn't affect the cell viability. Then they want to test what's the best length of exposure to have a good decrease of the percentage of the gene editing. And they assumed that the best was between 45 and 60 seconds.
So, once the parameters were set, they started with an in vitro analysis. So they transfected a cell line called EC293 with both sgRNAs or dual Brex sgRNAs. Then, after 2 hours, they disposed this culture to light up to 60 seconds. Both the population were allowed to recover for additional 2 hours, after which the RNA was harvested, and analyzed by a technique called digital droplet PCR. They found that the dual breaks RNAs exposed to light, shown here by the green column, exhibit a significant decrease in abundance in full-length products when compared to the paired population kept in the dark, explained here by the light blue column. This was not true for the standard sgRNA, as you can see here. Then they moved to test the ability of the CRISPR to modulate the genome editing events within human cells. So, 4 hours following the delivery of the standard sgRNA or dual breaks sgRNA targeting a gene called DNMT1 into the same cells that I mentioned before called EC293, the samples were illuminated up to 60 seconds and allowed to recover for an additional 44 hours. After that, they harvest the genomic DNA of these cells, and this DNA was analyzed using a technique called ICE or interference CRISPR edits that can detect the mutations like insertion or deletion caused by the CRISPR method. The results shown that the degree of editing was significantly reduced in light-exposed samples, as you can see here by the green columns, only for the dual breaks sgRNA, but not for the standard guide RNA. At this point, they wondered when they should start irradiating the samples to have a maximum efficiency of the CRISPR system. So, to reply to this question, they prospected another time they had 293 cells with dual breaks sgRNA targeting the DNMT1 gene and illuminated at distinct samples 1 time each every 2 hours for 2 days. After 48 hours, the genomic DNA was isolated from all samples and analyzed for the insertion deletion mutation using the ICE technique. As you can see in this graph, the CRISPR allowed to titratable levels of genome editing within samples. Seeing the results, they decided to set the letting time 4 hours after the transfection. To test the universal effectiveness of the CRISPR system, they create a panel of standard sgRNA and dual breaks sgRNA targeting a variety of chromosomes and local genomic content. They used the same design used before for this experiment, so they transfected the cells with both sgRNA or dual breaks sgRNA. Then they split both the population in irradiating or non-irradiating one, so they analyzed the level of editing in all these conditions. Across all targets, all but 2 intact dual breaks RNAs, shown here by the boxes, formed double-strand breaks at similar frequency as standard sgRNAs. The majority of the dual breaks sgRNAs also show a decrease in editing efficiency while illuminating 4 hours post-transfection, compared to cells from the same transfection that remain in the dark, as shown here by the light blue and green columns. Importantly, the irradiation did not decrease the editing efficiency of the standard sgRNA, suggesting that the incorporation of the photoclippable linkers was wholly responsible for the decrease in efficiency. The same experiment was performed also with other 2 cell lines called U2OS or Hep3B, and the results were comparable. Then they observed that one target called FANCF was inactivating to a lesser degree than others, and they hypothesized that the decrease in efficiency could be based on the individual editing kinetics at each site. So, to test this hypothesis, they transfected the cells with either standard or dual breaks sgRNA, and they were illuminated 50 minutes post-transfection, so earlier, and the DNA was isolated and at the indicated time points. In line with the hypothesis, the illumination of the dual breaks RNAs 50 minutes post-transfection, so earlier, completely ablated the editing at each site, as you can see here by the green line. Interestingly, at 4 hours post-transfection, they observed nearly 50% of alleles in the standard sgRNA transfection had already experienced a double-strand breaks that was repaired through a non-homologous enzyme method, confirming the hypothesis that the editing kinetics at each site is very fast. Another problem of the CRISPR technique is the binding with the off-target sites. So, the researcher at this point want to try to optimize the on-off-target editing events. To test this, they include a standard guide RNA known to be cytotoxic for the cells due to having an off-target site in an essential gene for the cell survival. That means that if this sgRNA coupled with the off-target site, the cells will die. In this image, you can see the nuclei of the cells marked in blue by the Hodge staining. Interestingly, where the dual breaks sgRNAs of the guide was used in conjunction with the irradiation, a greater portion of the cells survive, as you can see here by the upper part of the image. This is potentially due to an increase in the ratio of on-off target events while maintaining editing efficiency. Then, they started evaluating the ratio of on-off target editing at various time points post-transfection at the FANCF locus. So the on-target gene editing happens quickly, as you can see here by the solid line, and reach saturation, while the off-target editing lags before becoming saturated at later time points, as you can see here by the dotted line. The identification of these inflection points can minimize the harm of the off-target editing events. They created a single guide RNAs that had significant levels of off-target editing at 1 or 2 sites within the genome. They try to maximize the ratio between on-off target editing by illuminating the dual breaks sgRNA at an optimal time point post-transfection. Then, they transfected independent pools of cells with 7 unique sgRNAs and exposed the pools to light at anticipated time points. Then, they harvested the genomic DNA from each of these pools to form a longitudinal editing curve. The results show that following illumination, the degree of editing at many off-target sites plateau, as you can see here in the bottom part of the graph, described by the light blue lines, demonstrating that inactivating dual breaks RNAs slow down the off-target editing, as you can see here by the light blue lines. To better understand these results, they represented also these results in this table, in which the more is the off-target cutting ratio, the darker is the blue. So, the off-target effects were lower at the same endpoints in samples that were illuminated sooner after transfection. So, as you can see here, the samples that were illuminated 4 hours after transfection show a normalized off-target editing ratio higher than the other conditions, and also higher compared with the sgRNA, standard sgRNAs. The last part of this paper is focused on the spatial control of the editing by the CRISPR technique. In fact, one of the major advantages of using optical as opposed to chemical stimulus is the ability to obtain precise spatial control. To test this, they create a thin film mask with the darker areas, as you can see here in this image, that are transparent and allow light to pass through and deactivate the dual breaks sgRNAs. Then, the clear areas that are opaque, in which editing continues unimpeded. For this experiment, they use a GFP expressing cell line and design a single-guide RNAs to create a GFP knockout phenotypes. They use a standard inverted fluorescent microscope which could illuminate a single well at a time. This fluorescent microscope setup contains a 385 nanometers LED commonly used for illumination, and it is right on the edge of the reactivity of the photocleavable linker in the dual brake sRNA. In this picture, the blue dots are the nuclei of the cells, and the white is the GFP protein expressed by the cells. As you can see, the GFP is knockout, so it appears just blue, only in the part covered by the opaque mask that's blocking the light passage. On the contrary, the rest of the cells express the GFP protein because the mask protects the cells from the light, and the GFP was not cut and can be expressed by the cells, and the cells appear white. Concluding, the CRISPR allows for tight control of editing from both a spatial and temporal perspective, expanding the toolbox of optogenetic gene editing. Of course, not all glitter is gold. In fact, the major limit of the CRISPR technique is its application in vivo, due to the low penetrance of the UV light through tissues. That's all. Thank you all for your attention, and we will be happy to answer to your curiosity.
Good morning, everyone. I'm Giorgia. I hope you enjoyed our presentation. So now, with Ilaria, we will try to answer your question. So, okay. First question, Marco asks, do the authors compare their technology to the already implemented Cas9 mRNA sgRNA transfection? What are the advantages, disadvantages of the two approaches?
Good question. No, they don't compare this technology with other techniques.
Okay, thank you, Ilaria. Okay, Valeria asks, can it be useful in diagnosis or treating COVID?
Yes, with the arrival of COVID-19 pandemic, some researchers are experimenting with rapid tests based on the CRISPR-Cas12 system, and the presence of the virus can be detected in blood or urine sample using the CRISPR-Cas12 system linked to a fluorophore. And as Cas12 cues the viral RNA, fluorescence is detected.
Okay, thank you, Ilaria. Does anyone have any other question? Marco, do the authors think this technique can be applied also to CRISPR and CRISPR approaches with the Cas9 or nicases?
In the paper, they don't talk about this type of application, so I don't know.
Okay. Why did they have to use a specific wavelength?
Because the use of more penetrating wavelengths could induce damage to DNA or to other molecules of the cells.
Thank you. Professor Teti asks, can you comment on the likelihood the method will be used for therapy?
Yes, the researchers are experimenting the use of CRISPR-Cas system to treat some disease like beta thalassemia, cystic fibrosis, muscular dystrophy, and diabetes. And they could use CRISPR-Cas system in genic therapy to delete a gene or to repair the mutation that caused the disease.
Thank you, Ilaria. Does anyone have any question? Other question?
Sorry, Ilaria, maybe Professor Tati want to know what are the risks to use these methods for therapy. So if you know some risk about the use of this technique.
Yes, CRISPR-Cas could induce a double break damage on the DNA, or could induce damage near the sequences that you try to modify.
So some specific binding and cutting, right?
Yes.
Okay. No more question? Curiosity about it?
Also, Marco, if we have something about the YouTube channel.
Marco asks, do they plan or creating a CRISPR on version of this?
No, they don't plan a CRISPR-on version.
But it could be interesting. Very good. And question. Maybe it could be, I don't know, something similar for make it on. Yeah, it's very interesting. Sorry, I don't know if there's some technical problems with the audio. Okay.
Okay. Other question? Someone want to ask something? No. So
If when I read this paper and I study about it, I think about something that could be linked CRISPR and vaccines. So, Laria, do you read something about it?
Yes, for COVID in a specific way, the researchers are training to create a vaccines using adenovirus like vector, which the A1 replicating gene is deleted, and in place of this gene is inserting the gene corresponding to a surface antigen called Arpion of the SARS-CoV-2 virus, and through the action of CRISPR-Cas system. And in this way, the surface antigen stimulate the production of antibodies by the immune system.
Okay, thank you. Very interesting. Okay, I think we have no questions from the audience. If you want to understand better, please don't be shy right now. Otherwise, I think that we can thank all of you for the attention. And I don't know if Marco has some updates for the TMS, the next TMS meetings.
Yes, so this is the last TMS seminar of the year. So I don't know if Professor Teddy wants to say some, let's say, goodbye words before we restart next year. I don't know if her connection is good, but if it is,
why me?
because you're the chair.
You
of
can
the
say
TMS.
goodbye. Merry Christmas. Go ahead.
Okay, so thank you all for being here today and in all the TMS. We're always very numerous, so thank you so much for the support. And the next TMS event will be on the 7th of January. So have a Merry Christmas, Happy New Year, and see you next year, always in the same team, the TMS. Thank you.
Thank you all. Goodbye.
Bye-bye.
Have a nice holiday.
Thank you. Bye.
Thank you. Bye. Goodbye.
Goodbye.